Hello, horticulture students. This is Chapter 13, Seed Propagation, and I'm taking this information from the Horticulture Today textbook. All right, so we're going to consider these flowers on the screen. These are zinnias, and they are personally one of my favorites to grow in the garden. I like to do a row of them because they are just so easy to grow. I love how inexpensive they are, how brightly colored they are, and how well they typically grow. So zinnias are usually where people say, hey, if you want to start growing flowers from seed, especially cut flowers, start here. But sometimes people get overwhelmed and they start asking themselves a bunch of questions. Where do I start? What do my seeds need to grow? What if the seeds don't grow? How do you save and store these seeds? And it can be a little overwhelming, but the thing about horticulture and plants is just work with them. Try it, see what works. There's so many different resources out there to help you, but we're going to work through this chapter over the science of how seeds grow and how they develop, and then how to also plant them in a way that's going to give you the best success. So we're going to start off with seed morphology and development, and this is that science behind how the seed is formed. So something you need to know about the seed is this is the reproductive unit of the plant. It has to occur through the process of fertilization. So we have a male flower and a female flower. They fertilize each other. The female begins to make the seed and then the fruit that surrounds that seed. So it is a sexually reproductive process. And the seed is going to contain three things. We're going to have that protective seed coat on the outside of the seed. It helps protect that plant from environmental things like wind and cold and water and things like that. And then you're also going to have that endosperm, which is a storage tissue in the center of the seed. When seeds are in the ground, before they start to produce their own food through photosynthesis, that endosperm is going to contain the starches and things and lipids, proteins, oils that seed needs to grow. And then we're also going to have the embryo on the inside of that seed, and that's what becomes that baby plant. So here you've got an image of a corn seed that is a monocot. Monocot means that the plant has one seed leaf that comes out of the seed. Corn is an example. And then you have a bean seed example, and a bean is going to be a dicot. Dicots have two seed leaves that come out when the seedling germinates. Now, when we're talking about the different stages of how that seed is developed, we're going to have three different ones. Histodifferentiation is where the cells are beginning to divide and they're beginning to become their own characteristics. So there's different things that plants will need, different types of cells, and histodifferentiation is where that division and that cell expansion, not cell expansion, but the seed size is going to increase rapidly because of the division happening. And it's going to be where those leaves are formed and the cells that become the roots and things like that. So just remember differentiation, things are becoming different. In the cell expansion stage, that's where your seeds are going to really quickly increase in size. So the cell enlargement is due to accumulation of food reserves in the form of carbohydrates, fats, oils, and proteins. So these seeds are also going to need those things so that they can grow and have the energy they need while they're underground. Maturation drying is our third stage of seed development. And this is where it starts to become more mature. It's that last stage of seed development. It's the point at which seeds have reached physiological maturity, meaning they're not going to get any larger. They experience a rapid water loss because they're no longer connected to the mother plant. They are cut off from their water source, so they're going to begin drying, which is not a bad thing. That's a good thing. And they're basically going to desiccate because they're no longer they're basically drying out, and that's going to allow them to be quiescent and dormant. So a quiescent seed is going to be a seed that has dried out. If I give a quiescent seed water, it can germinate from that. A dormant seed, and we're going to talk more about dormancy later, even if I give that seed water, it's not going to germinate because it has some dormancy issues it needs to overcome. All right, so another thing I was going to mention on this slide we're talking about these stages of seed development. Typically, the maturation drying happens, the fruit around the seeds dries out, and then it becomes dispersed. But vivipary or vivipary, whichever way you want to call it, Google says it 2 different ways. This is a unique natural phenomenon, and it occurs in live people, animals, naturally. So if an animal gives live birth, this is what's happening. But for fruit, this is something that's crazy. Like, you see the picture of a tomato, and there are plants growing out of the tomato. So when we see the tomato, typically we just see the seeds, but these seeds have began to germinate before they are released from the fruit. So that is vivipary. The next word I'm going to mention, I'm not going to ask you to really memorize it. We're not going to go too in-depth on it because it's one of those things as a horticultural class. We're more focused on how to grow things and how to keep them healthy. And this is really getting into the physiological science of the seed. So apomixis, some plants are able to produce seeds spontaneously without going through the fertilization process. This is common in chickens. Chickens can lay eggs without a male, okay? Or they can lay eggs without a male. Hopefully I just said that right. Now, that does not mean they are fertilized. But with plants, plants can go through asexually and create a seed without fertilization having happened. So when apomixis happens, it's going to be asexual and it's going to be a genetic clone of whatever plant it came off of. Now, typically this happens more in your citrus fruits, and you can't tell when you go in the grocery store and you get a bag of oranges which might have been reproduced through apomixis or through natural sexual reproduction. So it's just one of those interesting things to kind of be aware of, is that some seeds can spontaneously occur asexually, but very rare. All right, so the next large section we're going to get into is what we need for our seeds to germinate. Germinate means basically to grow. This is where my seeds begin to grow. So I've talked about how the seeds were made. Now I want them to grow. So seed germination is the process by which they begin to grow, and the conditions, 3 conditions must be met in order for the germination to occur. So first off, that seed has to be viable. Viable means that the seed is alive. It's going to actually produce something. Number 2, environmental conditions must be favorable, so having the right amount of water, proper temperature, oxygen, and sometimes light, depending on the plant species. And then number 3, dormancy conditions have been overcome. And we're going to talk even more in detail about those dormancy issues later. So if these conditions have been fulfilled, then the early phase of germination can begin. And in early germination, 3 phases describe the process of a seed beginning its water intake. So this was the 3 requirements we need. This is the 3 phases of a seed germination. The first one is imbibition. This is where that plant is just going to absorb up water. Okay, imbibition, it's absorbing the water rapidly so that way it can hydrate its inner tissues. The second phase is the lag phase. Here it begins to slow down on the water that it's absorbing. This is a period where many cellular activities are preparing the seed to grow. So imbibition is gathered up all that water to hydrate itself. And then in lag phase, cellular things are starting to happen. And then in the third phase, we have radical protrusion. So a radical is that root that comes out of the seed. So protrusion is going to be it coming out. So now it kind of makes sense. Radical protrusion is the emergence of the seed root. Okay, you can see that beginning to happen. And then you're also going to see where that seed stem or shoot begins to emerge. We can also call that a plumule, Plumule. So here we can see the radical, which is the root, and the plumule, which is the shoot, starting to emerge. Now, once this seed has crossed that threshold for where it is at the soil level and it begins to emerge, then the seed leaves can start to begin absorbing sunlight and making its own food. So when we get into the germination, then a lot of times we want to talk about germination rates. And there's a few different words we're going to use, and they each mean something a little different. So they're going to have these variable germination rates, and they're measured by these indicators. So the percentage is going to be what percent of that seed lot, so a group of seeds, actually comes up. If I plant 100 seeds, 70 of them germinate and rise above the soil level. 70 out of 100 is going to be 70%. So my germination percentage is 70%. If I talk about my germination rate, I'm talking about the speed it took for that plant to germinate. So a lot of times on the back of seed packs, you'll see how many weeks it might take for seedlings to emerge. So 1 week or 2 weeks or 7 to 10 days. That's going to be the germination rate, the time or the speed. Germination uniformity is the time frame in which the seeds emerge. So with the uniformity, we want plants to come up at the same times, so that way they're more uniform in size, especially when we go to transplant them or sell them. So seed lot is that group of seeds when we're talking about a particular crop. So an example of a seed lot would be if I go out and I plant a tray of Better Boy tomato seeds. Okay, I'm going to plant all of those Better Boy tomato seeds at one time and in one thing. They're going to get the same kind of treatment. That is the seed lot. Okay. Some environmental conditions that I need for germination. The first thing is going to be the correct temperature. Until the plant has, or that seed is feeling the correct temperature, it's not actually going to grow. This is our most critical factor. Temperature influences seed dormancy control, germination percentage, and rate. Seed germination temperatures have been identified for most horticultural species, and they'll give us a minimum temperature and a maximum temperature. Some even give us an optimal temperature, so that way we know. So in a greenhouse especially, we usually try to find a temperature that's kind of in the middle of average of what we're growing completely in there, so that way we can set it at a temperature things are going to grow. Okay, so you do have different sets of plants. So you have species that prefer more cool temperatures. Okay, so those are going to germinate anywhere from 86 to 104 degrees Fahrenheit. If the temperature, that's if it's tolerant, excuse me, cold temperature tolerant. So 86 to 104 degrees Fahrenheit is typically pretty warm. So it's tolerant of that, it can handle it. Now, if it requires a cool temperature, it will not germinate if the temperatures are higher than 77 degrees Fahrenheit. So you're going to have to keep it below that in order for that seed to actually grow. The next set is going to be the warm temperature seeds. So these plants are not going to germinate at temperatures below 50 degrees Fahrenheit. This is things like asparagus, sweet corn, tomato. Some plants have to have it 60 degrees or higher, and that's going to be beans, eggplant, peppers, cucumbers. So those plants, if they are exposed to a low injury, they can show chilling injury, and it's basically stunted growth, or this stem doesn't look right, or it just doesn't grow. All right, so temperature was our first environmental condition we need. Water is the next. Okay, so temperature is the most critical because it's going to be what actually allows the plant to grow, but then water is what starts the germination process. Until water hits that seed, it's not going to begin to grow. So we have to have water. Now, there are some other things that we could talk about with water, like seed priming. Seed priming is when we basically soak those seeds in water, and that's going to hydrate it, and then we can dry it out. So that way the plants are going to grow all at a uniform rate. Okay, with plugs, these are going to be seeds that are grown in containers to transplantable size, and that's what's on the picture here. And we're going to talk about more about plugs later on when we talk about growing seeds in the greenhouse. But water is our next environmental condition. Oxygen and light. Plants have to have oxygen to be able to respire and produce its own energy. And then there are going to be some plants that want light, and there are going to be some that do not grow well in light. So it's important to know what species of plant you have and know what it requires as far as light and dark. For plants that love light and need a lot of light to germinate, you can plant them more shallow so more sunlight can reach them. For plants that like dark, we can cover them with a piece of newspaper, and that's going to help shade out the light. So then you can also get into talking about the light quality, how much light it's getting, and even light sensitivity. of that seed.